Hello everyone, this is Sapna from RPIIT Medical and Technical Campus. In previous class we have discussed about lipid chemistry, but in today's class we are going to discuss about the digestion and absorption of lipids. So first of all we will discuss about the digestion of lipids. Digestion of lipids involves triglycerides, phospholipids and cholesterol esters. Digestion, uh, Digestion of triglycerides involve glycerol and triacylglycerol, and they are digested as emulsification and enzymatic hydrolysis of lipase enzyme. First of all, we will discuss about emulsification. Emulsification is known as the breakdown of large fat globules into small ones, which occurs in mouth by chewing of food and then in stomach by peristaltic contraction, in intestine by peristaltic movement with the help of bile salt and lysophospholipid. Then we will discuss about lipase enzymes. Type 1 involves lingual lipase enzyme. Type 2 involve gastric lipase enzyme. Third one is pancreatic lipase enzyme. Fourth one is intestinal lipase enzymes. These all enzymes are most active in pancreatic lipase. So first of all we will discuss about lingual lipase. Lingual lipase is secreted by the dorsal surface of tongue, which is known as von Ebner's gland, is not of much significance in human compared to rat or mouse. Then we will discuss about gastric lipase. The pH of gastric lipase is 3 to 6. So triacylglycerols or short and medium chains of unsaturated fatty acids in presence of gastric lipase enzyme in infants are converted into short and medium free fatty acids and 1,2 diacylglycerols. Then we will discuss about the role of pancreatic lipase. Here we can see triglycerides in presence of peristaltic lipase, colipase, bile salts, phospholipase, calcium is converted into beta monoglycerides and two molecules of fatty acids. In presence of isomerase enzyme, beta monoglycerides are converted into alpha monoglycerides. Then in presence of pancreatic lipase, colipase, bile salts, phospholipids, calcium 6%, alpha monoglycerides are converted into glycerols and then fatty acids. Then we will discuss about intestinal lipase role in digestion of lipids. It acts as within intestinal mucosal cells and then hydrolyze the absorbed primary alpha monoglycerides, forming glycerols and free fatty acids. Then we will discuss about the digestion of phospholipids. Phospholipids are converted into lysophospholipids. In intestinal, phospholipase may complete the hydrolysis of lysophospholipids. Phospholipase A2 activate by trypsin and required bile salts for its activity. Then we will discuss about the digestion of cholesterol esters. Cholesterol esters by cholesterol esterase enzyme are converted into fatty acids and free cholesterols. Then we will discuss about the absorption of lipids. Lipids are the end product of the lipid digestion are monoglycerides, fatty acids, glycerols, cholesterols and lysophospholipids. So first one is glycerol and short chain fatty acids. It is water soluble carried through portal circulation. And then long chain fatty acids. These are monoglycerides, cholesterols and lysophospholipids needs bile salt to be absorbed. Then bile salt surrounds these components by their unpolar end while their polar ending directing outward, and it is converted into water soluble micelles whose size is 0.1 to 0.5 micrometer in diameter. Micelles soluble in water enter to microvilli of mucosal cells by endocytosis, where fat digestion may be completed through the action of intestinal lipase. And here we can see the diagrammatic view of lipid absorption. After that, in the mucosal cells, and triglycerides and other lipids are resynthesized once again. And here is the digestion and absorption of lipids is completed. Thank you. Have a nice day.